Okay, now it's recording. Vale, pues, bueno, gracias a todos por venir. Esta charla la va a dar Cláudia Norte, que es profesora, investigadora profesora en la Universidad de Coimbra, Departamento de Ciencias de la Vida, en el Instituto de MARE, digamos, Instituto de Investigación Marina y Ambiental. Parte de su investigación mayormente se ha centrado en la interacción ecológica entre la bacteria Borrelia y sus predadores, tanto finales como intermediarios, ¿no? Garrapatas y diferentes vertebrados, que será de lo que trate la charla. Y por otra parte, en los últimos años se ha centrado más en, digamos, en la parte de la ecología evolutiva o ecología pura y dura en aves, sobre todo en páridos, que también nos dará unas pocas pinceladas al final de la charla. Y ya por último decir, pues, que nosotros conocimos a Cláudia a raíz de una estancia internacional que yo estuve allí, me supervisó durante 4 meses, trabajo de campo con el río carbonero y parte de trabajo de laboratorio. ¿Vale? La charla será en inglés, ya os digo, contará básicamente la parte desde que terminó la tesis hasta ahora y las últimas pinceladas sobre sus últimas líneas de investigación. Okay, you can start when you want.
Hi. Good afternoon. Thanks, George, for the presentation and for the invitation. I would like to thank you and Gregorio. Today I will talk about the work I've done since 2010 on the ecological interactions between the bacteria Borrelia, that is the etiologic agent of Lyme borreliosis, and its hosts, either vertebrates and the vectors, the ticks. This work was done in collaboration with the National Institute of Health in Portugal, but also some European institutions in Germany and Switzerland. So, for long, wild birds have been known to play a role in zoonotic diseases because they are reservoir hosts for some microorganisms that are pathogenic to humans. And birds, especially the migratory ones, may disperse not only the pathogenic agent, but also its vectors. In the case of vector-borne diseases, especially those transmitted by ticks. So they may amplify the pathogens locally or transport them to new locations, creating new foci of disease. So they contribute to frequent, fast, and long-range dispersal of several pathogens. And already several threats to public health have been associated with birds, including several viruses, such as influenza A virus, West Nile virus, Crimean Congo hemorrhagic fever virus, and some bacteria, such as Rickettsia and Borrelia. So because bird movement knows no country borders and continental borders, there is a need for international joint efforts of surveillance of these diseases associated with birds in order to better prevent its spread. So, Lyme borreliosis or Lyme disease is caused by Borrelia burgdorferi sensulato, which is a bacterial species complex that includes more than 20 species. And is a multisystemic disease affecting the dermatological system, the cardiac system, neurological, and joint. It has also joint manifestations. It is transmitted to humans by the bite of the hard tick Ixodes ricinus in Europe, and is the most common vector-borne disease of moderate climates of the Northern Hemisphere. And it can be considered an emergent zoonotic disease because new species are constantly being discovered, and they have been detected also in new areas. So these bacteria are maintained in nature by reservoir hosts, which are vertebrate hosts that are common and commonly infested by the vector tick. They allow the survival and amplification of the bacterium in their tissues, and usually they don't show any clinical symptoms of disease. And they are significantly involved in the infection of the vector tick. And already several species of birds, lizards, and small mammals have been proved competent reservoirs for Borrelia. So our first goal was to understand the importance of birds in Borrelia's zoonotic cycle over a broad geographical range. So one way to do this is to collect ticks from birds and check their infection status by Borrelia. And we did this in 10 European countries, which are in light blue in the map, by capturing birds with mist nets and in the nest boxes. And we collected over 2,000 ticks from over 800 infested birds. The most common tick collected was Ixodes ricinus, the generalist tick that is the main vector tick in Europe. But we also collected some ornithophilic ticks that mainly feed on birds, such as Ixodes arboricola and Ixodes frontalis, and some other species, but in smaller percentages. We found out, this is data from Portugal only, we found out that foraging behavior of birds affected their infestation levels by ticks, and those that foraged for food in shrubs or low vegetation had higher prevalence of infestation by ticks and higher intensities of infestation. That was the case of robins, blackbirds, wrens, etc. Regarding the infection status of those ticks that they carried, at the European level, we found infection levels of 37%, more or less, and in Portugal, it varied between 10 and 20%. But not all ticks from different bird species were infected in the same level, and ticks coming from thrushes like Turgis pilaris and Turgis merula were usually more infected than ticks coming, for example, from the robin. The robin carries many ticks, but usually they are not infected. So the effect of bird species is very important in this Borrelia cycle. Not all birds contribute equally to the maintenance of Borrelia in nature. The diversity of Borrelia species in the ticks from birds was relatively high. We found 6 species of Borrelia, and the main one, the more prevalent, was Borrelia garini, which is a neuropathogen to humans, followed by Borrelia walaisiana, and we also found some other species, such as Borrelia turdi, and even one new Borrelia species that hadn't been described yet, which we called Borrelia alligera due to its association with birds. Borrelia turdi in this study was found only in Portugal, and it was the most prevalent in ticks coming from birds. About Borrelia turdi, it had been detected in about 1996 for the first time in Japan in an ornithophilic tick, Ixodes turdus, feeding on Emberiza spodocephala. And only in 2010, it was detected again in European countries, first in Norway, and then Belgium, Spain, and Portugal. It was detected in different bird species and in different ticks, ornithophilic ticks feeding on them. And the main ornithophilic tick infected with Borrelia turdi was Ixodes frontalis, especially in Portugal. So, for example, this Borrelia species, has been detected in more countries recently, and in new vertebrate hosts and in new vector tick species, so it can be considered an emergent species. However, we still don't know its pathogenicity to humans. So one way to study the role of different vertebrate species in the cycle of Borrelia is to collect ticks from them and analyzing their infection status. Another one is to do some in vitro studies. There is knowledge that there are some species like Borrelia garini, valesiana, and burgdorferi sensu stricto that are associated mainly with birds, and others like Borrelia afzeli, which also is pathogenic, is mainly associated with small mammals as hosts. And one way to researchers found this is using those in vitro studies in which they expose different Borrelia species to the serum of these vertebrate hosts and check if they survive after incubation with their serum. Because in the serum there is the complement system of the immune system, and the complement system is thought to be very important in defining the reservoir competence of vertebrate hosts for Borrelia. So we captured birds of different species. We captured robins, blackbirds, and also great tits, and collected blood from them and also from race pigeons, and obtained their serum and exposed 4 species of Borrelia to the sera of birds. And after incubation, we checked if those Borrelia were viable or not. And these were the results. And for the majority of combinations, Borrelia garini, valesiana, burgdorferi sensu stricto, and afzeliae, most of them survived in the presence of bird sera, with the only exception of the combination of Borrelia afzeli with blackbirds. After 6 hours of incubation, about 30% of the spirochetes had died. So this means that they cannot survive in the blood of the blackbirds. These results are mostly in accordance with what was known, because we already knew that Borrelia garini, Borrelia valesiana, and sensu stricto were associated with birds. But most of knowledge said that Borrelia afzeli was associated with small mammals and could not survive in birds. But these in vitro studies show that it may survive until a certain point in birds' blood. So the results cannot be generalized between Borrelia strains and different bird species. So it's possible that Borrelia afzeli survives in birds' blood, and if birds are not competent for, except the blackbird, if birds are not competent in nature for Borrelia afzeli, it has to do with some other components of their immune system, and not the complement. So the blackbird, it seems like a very important bird species in the cycle of Borrelia, because it's very common. It inhabits urban and rural habitats. It's commonly infested with ticks, and the infection of Borrelia in its ticks, it's very high, well, from 20 to 58% of infection. And we already had isolated spirochetes, Borrelia, in skin biopsies that we had collected from the blackbird. And one way to definitely prove that a vertebrate species is a competent reservoir for Borrelia is to do a xenodiagnostic experiment. And that's what we did with blackbirds. This means that we captured blackbirds in an enzootic foci of Borrelia. So these birds already had contact, probably they already had contact with Borrelia and with infected ticks. And we captured them, we took them to the laboratory, and we put larvae of Ixodes ricinus that were bred in a laboratory colony to feed on them, on the birds, and then we wait for them to feed, to molt into nymphs, and then we test those nymphs for infection. And we had 5 blackbirds in our experiments, and their tissues were tested for Borrelia as well, and 2 birds had skin biopsies infected with Borrelia valesiana, and 2 birds had Borrelia turdi infection in their tissues. And from these birds, the xenodiagnostic ticks, 20% were infected with Borrelia valesiana, and about, I cannot see it here, but I think about 5% were infected with Borrelia turdi. So this shows that blackbirds are able to maintain those 2 Borrelia species in their organism, transmit them to feeding ticks of the main vector tick of Borrelia, feeding larvae. These larvae maintain the infection through molt, and then if they feed in another host, they may transmit those Borrelia to the new host, which may be a human. So it proves the reservoir competency of blackbirds for these 2 genus species of Borrelia. So we already knew that there were some Borrelias that were associated with birds, and some of them circulated only in birds and their ornithophilic ticks that fed preferentially in birds. But we wanted to know, because birds are also infested by the generalist tick, that is the vector of Lyme borreliosis, we wanted to know if this generalist tick could acquire also those species, Borrelia species associated with the birds, and potentially transmit those species to other hosts, bridging those cycles of birds and their ticks to other hosts. So we did a transmission experiment with an ornithophilic tick, Ixodes frontalis, and a generalist tick, Ixodes ricinus. We again captured blackbirds in the wild, and we put larvae of those 2 tick species feeding on them, because we wanted to obtain nymphs that were potentially infected with the species of Borrelia that circulated within blackbirds. After obtaining those nymphs, we put those nymphs, potentially infected, in naive blackbirds that were bred in captivity and which were negative for Borrelia because they were never exposed to Borrelia. So we put those challenging nymphs feeding on them, and after that we did a xenodiagnostic again and checked if those birds, naive birds, acquired the infections from the ticks. Additionally, we checked the health status of those naive blackbirds before exposure and after exposure to Borrelia and Borrelia infected ticks. So these were the results. In the left part of the graph, you can see the infection levels we obtained in the challenging nymphs. So we obtained infections of around 20%, infections by Borrelia of around 20%, and those nymphs were infected, Ixodes frontalis were infected with 3 genuspecies, Borrelia turdi, Valesiana, and sensu stricto, and the Ixodes ricinus ticks were infected with Borrelia turdi and Valesiana. On the right part of the graph, we can see the results of the xenodiagnostics before exposure to the infected ticks and after exposure to the infected ticks in the naive blackbirds. And in the beginning, they were not infected. The prevalence in the xenodiagnostic ticks was 0, but after the exposure to the challenging nymphs, we had xenodiagnostic ticks in these birds infected. So the birds that were exposed to Ixodes frontalis acquired Borrelia turdi. So Ixodes frontalis can vector Borrelia turdi. The birds that were exposed to Ixodes ricinus acquired both Borrelia turdi and Borrelia valesiana. So this shows that Ixodes ricinus, the generalist tick, can vector not only Borrelia valesiana, which we already knew, but can also transmit Borrelia turdi, that genus species that was mostly associated with birds and their ornithophilic ticks. But as we found, it can also be vectored by the generalist tick Ixodes ricinus. And eventually, if these Ixodes ricinus were to feed on another host, such as a human, they could transmit these species to the human. So it remains to be known if Borrelia turdi is pathogenic or not. As I said, we wanted to know the effects of Borrelia infection in the reservoir hosts, because a reservoir host usually doesn't have much symptoms of infection, and there are also limitations to the harm that a parasite can do to its reservoir host. Although Borrelia is not really specific parasite, but in any way, if it affects the fitness of its hosts, it may also affect its maintenance in nature. There was no evidence that Borrelia reduced reservoir host fitness, but some physiological effects had been already detected in other studies, such as body mass changes, increased nitric oxide levels, and some immunological responses. So, as I said, we evaluated the health status of the naive blackbirds in the previous experiments before exposure to infected ticks and after, with some physiological parameters that reflect immunology, oxidative stress, and general health status. And what we found was that the effects were mainly dependent on sex of the birds, because we had males and females in the experiment. And most of the effects were related with oxidative stress levels. For example, we found that infected females compared with control females increased their glutathione peroxidase activities, which is an antioxidant enzyme. And for example, the infected males comparing with controls, they increased their protein carbonyl levels, which shows higher damage to proteins related with infection by Borrelia. So, because we found that Borrelia affected the oxidative balance of birds, it may impose non-negligible physiological costs on reservoir hosts, because we know that oxidative stress is responsible for cell senescence and aging. So if those infections and those effects occur in sensitive periods of the bird's life cycle, such as reproduction, it may really affect the fitness of the birds. And eventually the Borrelia maintenance in nature, in circulation. Okay, so as you remember, we had those ticks from birds all over Europe that were mostly infected with Borrelia garini. And we wanted to study the population structure of these Borrelia species. So we genotyped 8 housekeeping genes of our positive Borrelia garini samples, which were ticks from birds, and compared those the allelic profiles with other samples collected in several parts of the world, not only in Europe, but also in North America and in Asia. And we did a GOE burst analysis in which the allelic profiles are compared and the ones that are more similar clustered together. So each circle is an allelic profile, a sequence type. And those that are connected, they are more similar to those that are not connected among them. So what we can see is that Borrelia garini shows some divergence between among continents, because sequence types from different continents generally form different clusters. But in Europe, which are in the colors of blue and green, there is no apparent structure according to country of detection, with the only exception of 4 sequence types in gray rounded by a circle, which were detected only in Norway, in a remote area of Norway. There were only 3 sequence types that were detected in more than one continent, which are represented by arrows. So this is probably related with the dispersal movements and migratory movements of birds that lead to a special mixing of strains that are more often at a continental scale than a transcontinental scale. But even so, there are some identical Borrelia garini sequence types found in different continents, which is possibly due to long-distance migratory movements of some birds, as the seabirds. Actually, there is also a so-called Borrelia garini marine cycle, which involves seabirds, an ornithophilic tick that infests seabirds, which is Ixodes, and which lives in their nests or nest burrows, and completes its life cycle when the birds are in the islands breeding. During their breeding, also the ticks complete their life cycle. And although these populations of ticks, Ixodes, show a relatively high population structure, which has to do with the seabird species that they infest, even when those species breed sympatrically. There is no such structuring of Borrelia garini, because identical sequences of the flabby gene of Borrelia garini have been found in both polar regions. So there's some transhemispheric dispersion of Borrelia garini through seabird movements. And additionally, because when birds migrate, this is a stressful period for the birds. The mechanism of stress inhibits their immune system and leads to spirochemia, so higher levels of bacteria in their blood, and promotes the infection of ticks. Okay, so in Portugal, the main Borrelia species detected in questing ticks, the ticks that are found in the vegetation looking for hosts, is Borrelia lusitania. So, but in birds, we didn't find many Borrelia lusitania infected ticks. So we wanted to know which were the hosts that maintained Borrelia lusitania. This is a Borrelia species common in the Mediterranean area, but which has a patchy distribution in Central and Northern Europe. In some regions, its prevalence in questing ticks can reach 75% of infection levels. And we already knew that the lizards might play a role in the maintenance of Borrelia lusitania, because in Tunisia they already had done xenodiagnostic experiments with Psamodromus algirus, which is the large Psamodromus, and they found that this species was a competent reservoir host. So we did a focal study in Tapada de Mafra in Portugal, which is an enclosed game area with many ticks, because they have large populations of game animals, which are important tick reproduction hosts. And Borrelia lusitania is the most common Borrelia in the questing ticks. So another way to infer about reservoir competence of vertebrate hosts is to compare the infection levels of their ticks with those in the vegetation. Because in Borrelia and Ixodes, the larvae, when they are the Ixodes larvae, when they hatch, they are not infected, and they only acquire infection when they feed on an infected host and reservoir host. And then they will maintain the infection throughout molt. So if we compare the infection in larvae from a vertebrate host, and we found that there is infection by a certain Borrelia species in those larvae, probably it was acquired from the host, because larvae are not born infected, and it is the first blood meal of those larvae. So we wanted to do a study including several vertebrate groups. So we captured birds in mist nets, we captured small mammals in Sherman traps, and we captured lizards with a noose or by hand. And we captured questing ticks from the vegetation with the flagging method. And those are the results of the questing ticks. As you see in the first line is the infection levels of larvae of Ixodes ricinus, and they were not infected, but the nymphs were already infected because already had one blood meal, and the main infection was by Borrelia lusitania in a prevalence of 3%. Not very high, but still was the most prevalent species in the questing ticks. So when we compare the infection in questing tick larvae and larvae feeding on the blackbirds, for example, we see that the infection levels in the ticks from the blackbirds is higher for Borrelia valesiana, garini, iturdi, which is all in accordance with what we already discussed here, because the blackbird is known reservoir for these species. We also found 2 ticks from Apodemus sylvaticus, 2 larvae infected with Borrelia lusitania, but because these infection levels were so low, we cannot say that it has a prominent role in the maintenance of Borrelia lusitania. But if we look at the infection levels in Psamodromus algirus, we found that the levels of infection in its larvae were relatively high and higher than the questing larvae. So it had prevalences of 16%, 17% almost. We can do the same exercise for nymphs. Of course, needs from the vegetation already have some infection levels, but if you compare those with the nicks collected from Psammodramus algirus, Lacerta scraberi, and Podarcis hispanica, we can see that infection levels of those ticks are higher than those from the vegetation. So probably Lacertidae of those species play an important role in maintenance of Borrelia lusitania. About the population structure of Borrelia lusitania, you may imagine that it may be very different from Borrelia garini, because Borrelia garini is mainly maintained by birds which have large dispersion ranges, but lizards have small dispersion movements. So the population structure of the Borrelia they carry is likely to be very different and very structured. And it's what we found. We also genotyped 7 housekeeping genes of Borrelia lusitania in Portugal and some European countries where it has a patchy distribution. And in this dendrogram, we can see that we clearly have 2 clusters of Borrelia lusitania, one including species from North Africa and Portugal to the south of Tagus River, and in red, another cluster with samples from Central Europe and North Europe and to the north of Tagus River. This is an analysis with the same samples, but going burst analysis. And we also can see that the clustering pattern is similar. We have lots of connections of samples of Borrelia lusitania south of River Tagus with those of North Africa, and some connections of the samples of the north of the River Tagus in Portugal and Central European countries. And also, in accordance, a neighbor net with those samples shows that in one branch, the left branch, we have samples from North Africa and Portuguese isolates, and to the right, in the lower image, we have samples from European countries and only some Portuguese samples from North Portugal. So I talked to you about the importance of hosts in shaping the structure of Borrelia, but there might be also a role of the vector, because in this specific case, it was recently found a new species of Ixodes, which is very similar to Ixodes ricinus, but is only found in Mediterranean area and in some areas of Europe, very patchy. So we characterize genetically the ticks where we found Borrelia lusitania, the same ticks that I showed you before in the previous analysis. And one of the genes shows clearly 2 clades, one including Portuguese and North African Ixodes sequences, and another including the European non-Portuguese samples. But this doesn't explain completely the population structure of Borrelia lusitania, especially at that difference between North Tagus and South Tagus. So, going back to the host, Psammodramus algirus is a proven reservoir host of Borrelia lusitania, is widespread in North Africa and on the Iberian Peninsula, but a distribution very restricted in other European countries. Actually, in those other European countries where Borrelia lusitania is found, other lizard species play a role as reservoir hosts, and those lizard species do not occur in Africa or the Iberian Peninsula. And specifically for Psammodramus algirus, some studies have found that in the Iberian Peninsula and North Africa, there are differentiated populations between East Iberia in squares, and West Iberia, North Africa, represented by circles, and in the population of the West, there's some lineage differentiation between the north of Portugal, south of Portugal, and North Africa. So the role of the host in shaping the Borrelia lusitania structure may also be important, but this requires further studies. So future studies is to understand how narrow is the niche for reservoir hosts of Borrelia lusitania, using either transmission experiments or in vitro studies of serum sensitivity assays. Do a parallel genetic characterization of Psammodromus algirus hosts and Borrelia lusitania in the ticks they may carry in different regions of Portugal and Spain and North Africa. And also a special interest in studying the marine cycle of Borrelia burgdorferi, namely Borrelia garini, studying the infection prevalence and genetic characterization of Borrelia in ticks of different seabird species in different locations. Well, additionally, just to say that apart from this work that I have done until recently, I'm also focusing on the effects of stressful conditions on both the health of terrestrial birds and seabirds. Those stressful conditions can be contaminant exposure, human disturbance, and food limitations. And for that, I'm using some ecophysiological tools, studying oxidative stress and anabolic condition. And I'm also using non-invasive biologgers in the form of egg dummies that we place in the nests of incubating seabirds, in this case gulls, to register their heart rates and to study their stress responses in relation to human disturbance and other stressors. And at last, I thank all the people that worked with me so far. And without those persons, it wouldn't be possible to perform this amount of work. And also to the financing institutions. And for your patience to hear me out.
Okay, thank you, Claudia. Voy a parar de grabar.
Thank you.